Our next speaker is Jeff Toretsky, and he'll be talking about vincristine with YK4279 Ewing Sarcoma. Thanks, Jeff.
It's a pleasure to share this work with everybody. And I decided that I would share a little bit of our work, talking a small amount about the drug. You've heard a little bit about some of that so far, and some of the work that we've done now to try to take that drug and extend it further. So you've already heard Ewing sarcoma quite a bit. This is sort of my general Ewing sarcoma slide. It's named after Dr. James Ewing, who in 1921 identified this tumor because it was more sensitive to radiation than the other bone tumors at the time, which was osteosarcoma. One of the things that is fascinating about this, which Ewing has already spoken about and I could alluded to, is this chromosomal translocation. And you see here the normal chromosome 11 and the normal chromosome 22. And in Ewing sarcoma, there's this switch between that. And 95% of Ewing sarcomas have these two chromosomes, or very related, break and trade places. And it's this breaking and trading places that gives rise to a totally new protein, this EWS-Fly, which is only present in the Ewing sarcoma cells. So for many, many years, people thought, well, this is a target. And there were even articles written about how this had been known for 5 years, 10 years, 15 years, 20 years, and yet there hasn't been a drug that specifically targets the one thing that we know is critical for the development of Ewing sarcoma. And so through a lot of different, what I call laboratory party tricks, we could eliminate EWS-Fly, and we knew that it could kill the cells, but none of those had been really able to be translated into a human use. So some of the challenges to targeting this fusion protein was this sort of lack of protein structure to design inhibitory molecules. And ICUT gave a really good introduction to our sort of unconventional approach of doing drug discovery in our lab. And it turns out that using this BIACOR, this surface plasmon resonance technology, in a talk that I gave here last year describing how we got to this point, we can now tell you that TK216 is the analog of the original molecule we discovered called YK4279, and this is now in clinical trials, and I'll tell you about that a little bit more in a moment. One of the other challenges that people often throw up in front of me, they said, well, it's such a rare cancer. There's 400, 500 cases in North America each year. You're never going to be able to get anybody with any sort of commercial development. And of course, we found that. We actually found that a lot of the bigger drug companies weren't interested in supporting development of a drug for such a rare cancer. So I was sort of left with this option of starting a company, and the first company we founded called Targeting Disordered Proteins, it failed. I've heard that's actually a good sign to have your first company fail. And the second company we founded called Oncternal Therapeutics with a guy named Scott Glenn, who's a serial company starter, if you will, in San Diego. Is a great story behind that. I can tell you some other time. And he was able to go to his Rolodex, and I had the molecule, right? I had this compound we wanted to get into clinic, and he went to his Rolodex and in 6 weeks raised $3 million, and we're off to the races. And my disclosure is that I'm a co-founder of this company, and I have an equity interest in this. And the other thing is, even though people had done a lot of work with EWS-Fly, really understanding how it works we're just at the beginning. And one of the things that CCF is now supporting through Force 3 is understanding in a deeper way how EWS-Fly is actually working. And so we've actually spent some time figuring out that EWS-Fly is involved in this process called alternative splicing, and I'll talk a little bit about that today. So just to give you the 101 biology of Ewing sarcoma, most of you have seen this already. This is the chromosomal translocation. It leads to a fusion message, which gives rise to this fusion protein that has half EWS and half of Fly1. That, up until recently, was known to basically bind to DNA and turn genes on and off. We know that splicing is also involved now. And our goal was to say, well, could we find a small molecule that would stick to EWS-Fly and block its interaction with a critical partner? And when we started this, CCF was critical to our initial work because we had no preliminary data. We had this crazy idea that if we made EWS fly in bacteria and purified it, that we could do a screening like I could describe, where we would find a chemical that would just stick to this. And it turned out that one of the chemicals that we thought looked good, and by looking good, I mean it had pharmacologic properties that we thought we could use to go on to make a drug, and it had a lot of chemical openings for us to change the perimeter of the chemical with the central core of the chemical, what we call the backbone, remaining the same. And we ended up finding one that did block this protein called RNA helicase A. And of course, we did that again with the surface plasmon resonance. One of the things we were struck by that we published in our early paper in Nature Medicine was that this chemical would rapidly lead to apoptosis in Ewing sarcoma, and it was doing so at about 500 to 1,000 nanomolar. But as a control to see, well, how specific is this drug going to be? You know, how well is this drug going to really work? Can we amp up the dose, still get the apoptosis, obviously, in Ewing sarcoma, but we treat a series of non-malignant cell lines. These are keratinocytes here, these are endocervical cells, and these are, these are endothelial kidney cells, these are keratinocytes. And what you see stained here is the nucleus. And when cells undergo this death process called apoptosis, the nucleus fragments into these little bits shown by the arrows. But you can see that at this dose, which is significantly higher than the dose that it would normally cause to get this in Ewing sarcoma cells, we're able to show that in these non-Ewing sarcoma cells that they weren't affected. We've published a couple of papers with YK4279 demonstrating its action. And so as I mentioned now, this analog of YK4279 is now in phase 1 trial. What is a phase 1 trial? It means that we're looking for the dose and we're looking for human toxicity. And I can tell you with great excitement that we've now enrolled 14 patients. We're at dose level 5, meaning that we've doubled the dose 5 times. And there are there are 4 more patients in the queue that have not yet started, but they look like they're going to go on study in the next few weeks at dose level 5. And we're hoping by either the fall or certainly by next spring at this time, I can tell you a little bit more about how that study has gone and maybe there's some results. So we thought early on that no drug in cancer has been particularly successful as a single agent. That's even being proven true for the immunotherapy drugs. Things tend to need to be, you have to give cancer the old 1-2 punch. And so we asked, well, what would be the 1-2 punch that we could give with this drug? And could we identify mechanisms for apoptosis by knowing what drugs would be synergistic with YK4279? So in other words, we're looking A, for something that's going to be better at killing cells than either drug alone, and B, can we learn a little bit about how the drugs are actually working through this process? So we did a screen, and you can't see this because it's basically illegible, but I just want you to show there's a sort of color bar here, and the color bar was basically our way of scoring what the interactions would be like. And it turns out that in the reddish colors are the synergistic, the ones that act together, and one of the things that was fascinating was all of the drugs in a certain class, the vinca alkaloids, were acting synergistically with YK. Now what's also interesting is there's another class of drugs that we know are like the vinca alkaloids. They bind to the microtubules like the vinca alkaloids, but they do the opposite of the vinca alkaloids, and those are the taxanes. And what was interesting is those were not synergistic. So it had sort of an interesting in here, and we wanted to see, would the vinca alkaloids be synergistic with YK? So we actually did many, many experiments. A postdoctoral fellow who's now back running his own lab in Münster, Germany, Stefan Zellner, we didn't want to just do this experiment once. Every dot represents a repeat, and you can see that the vast majority of the dots for these 3 compounds fall below the line, which is where synergy is, and a few were above it, but the vast majority, statistically, we were looking at something that was very synergistic. And when we averaged all these together across a number of Ewing sarcoma cell lines, it looked like there were a group that were very synergistic for vincristine. So we went forward and we said, well, what about in animals? So it turns out In a lot of our early animal studies, we've had a lot of trouble with giving YK because of its solubility. But here, we were able to actually give very low doses of YK. You can see the YK alone at 50 milligrams per kilogram in the mouse really didn't do much over control. And the vincristine, which is the solid blue line, the vincristine alone was the solid black line, did improve survival a little bit, but not a whole lot. But the combination of the vincristine plus the 50 made a significant increase in survival compared to the control, which is in gray, which you don't see well because it pretty much overlaps with the YK alone at 10. So we were pretty excited by this. So when we looked at the actual tumors from these animals, we treated them for 3 days, just 3 days, twice a day, and harvested them all at the same time so we could actually get a sense of what is the tumor doing, what is the drug doing to the tumor cells before they would essentially disappear. And what you can see here is we saw these sort of donut cells, which were really interesting. And I showed this to some pediatric pathologists, and they said, well, that's really interesting because we saw the same thing in patients that were being treated with vincristine alone. And you can see this is in vincristine, but we saw more of them in the combination. So when we stained these cells with a process called tunnel, which tells us which cells are apoptotic, you can see that there's a couple of apoptotic cells in the YK alone. There's a few more in vincristine, but there was a significant increase in these apoptotic cells in the combination. So remember I mentioned one of the things we wanted to get a sense of is mechanism. So I'm going to show you 2 slides that give us a little window on mechanism. We don't have a lot of time to go into this, but one of the things we knew, and I mentioned earlier, was that vincristine binds to tubulin and messes up how it causes cell division. And you can see here, these are normal cells, and this is beta-tubulin staining in green. You can see that when we treat the cells with vincristine, we get this kind of chaotic pattern. The tubules form these little clumps, and the cells don't divide very well. Here's an example of a perfectly normal cell in the control undergoing mitosis. You see the chromosomes are lined up at the spindle, and you can just imagine if we had not killed that cell to take the picture, the cell would have continued to go on to divide. Well, when we stain the cells with a second agent that identifies gamma-tubulin, which normally only occurs at these spindle poles, and you can see that because the gamma-tubulin is in red and the beta-tubulin is in green, and you see where the yellow is, and you see how the combination of these is sort of overlapping, and the cell, and the cell is just about to divide at this point. Well, you can see that when we add vincristine, things are not that different, but when we add the 2 drugs together, we see a large number of these punctate lesions, and these are showing that not only are the beta-tubulin itself, but also gamma-tubulin, and there's been this chaotic changes in the actual cell division, which we believe then is going on to lead to apoptosis. A second mechanism that we've begun to look at this is through splicing. And one of the things we found is that in Ewing sarcoma cells, there are 5 variants of this protein called MCL1, which is a survival protein. And without having a lot of time to go into it, what I can show you is that when we treat cells with YK alone, we get this subtle shift where the MCL1 goes from this version with exons 1, 2, and 3, and we lose exon 2. And it turns out that when you lose exon 2, the cells are more susceptible to dying. And one of the things we showed is that over time, we're able to actually show that the level of MCL1S, which is the permissive of apoptotic form, goes up in the combination of, where are we going here? I'm sorry, the cells are dying more because of the combination of YK and vincristine. So in summary, our combination screening identified these vinca alkaloids as a class of synergistic compounds. The YK4279 has driven this alternative splicing. I showed you MCL1. We also saw this with BCL2. Vincristine synergizes with YK4279 through its blocking the microtubules and leading to apoptosis. And hopefully future clinical trials would then consider combining TK216 with vincristine. This work required a lot of people, and all sorts of different backgrounds and expertise. I want to just point out that Milton Yali were involved in the original synthesis of YK. Some of our work, and I'll just mention this in the introduction later today, was done in collaboration with David Loeb at Hopkins, which I didn't show you data for. And ICUT has been incredibly helpful throughout this whole process. Stefan has helped us wrap this work up, and we're now trying to get it finalized for publication. Saravana, Calliope, Verda, Haider, Garrett, Jessica, Grego, all helpful. Sun Hyuk did these early animal studies. And I also want to point out this CCF throughout all of this has been just critical because they supported all these ideas before we ended up getting all this NIH and Burroughs Wellcome funding. And for those of you who are interested, I want to point out a really fun event that's sponsored by Force 3 that raises money solely for CCF. On July 13th at the Jetty, there's this event called Crushing Cancer, and we went last year. It's great fun, so put it on your calendars and come on out and crush some crabs and have a good time with us. So thank you very much. It's interesting. We did do some timed experiments, and it looks like there is sort of a sequence timing thing with YK and vincristine, for example. But it's really hard in cell culture to get a sense of that because the cells are starting to die within 24 hours, and so you're starting to slice. It's very tricky. One of the things we're challenged by is this sort of sequencing. Is it the splicing? Is it something happening with the microtubules? There's a number of other genes that are regulated by EWS-FLI that are affected by the drug. So, one of the good news is that we're targeting this, what we think is really the vertex of where some of the, survival of Ewing sarcoma comes from. The bad news is that obviously sprays out into a lot of different areas, and trying to understand how any one particular piece of that kills the tumor cells is kind of tricky. Jerry.
Jeff, you mentioned immunotherapy, drugs, and treatments, and just following the mainstream media about cancer research, immunotherapy is absolutely the hot thing, at least in terms of a lot of the press. My question is, does immunotherapy have a promise for cancers that are more based on chromosomal abnormalities like Ewing sarcoma? Or are they different?
That's a great question. So it turns out recently, a drug called Keytruda was given FDA approval for a subset of colon cancer that had something called mismatch repair. So these are tumors that by their very nature generate a lot of chaotic chromosomal breaks and things like that. And it turns out that the immune therapy drugs in general have had their most clinical benefit has occurred in patients with tumors that have these very chaotic genomes, lots of errors, lots of broken, fragmented chromosomes, gene pieces, all sorts of things like that. And people have started to think about these as being potentially these neoantigens, and that's what this immune therapy is successful at. So flip that over now. There's a lot of sarcomas that have these simple, relatively quiet genomes, like Ewing sarcoma, like certain liposarcomas, a lot of other synovial sarcoma, although push that aside for a second because that actually might have some benefit in immune therapy, so I shouldn't have mentioned that one. But the point is that some of these sarcomas with these translocations and others are genomically quiet, and so far in the early trials that have been done, there hasn't been a lot of excitement for that approach for those types of cancers. Now, that doesn't mean there won't be, but it means that it's less likely based on what we now know, and so we need to keep doing things like this. Yeah.
Just wondering, P53 status with Ewing sarcoma is important as far as prognosis. Does the YK drug, is it dependent on P53 for the apoptosis pathway, or have you seen differences in the cell lines or data that way?
It's a great question, and first of all, P53 is only abnormal in a very small number of Ewing sarcoma patients. I think in the 3 studies that sequenced P53 and published it about a year ago, it was 10% or less. Arong, does that sound about right? 10% or less for P53 mutations in Ewing sarcoma patients? Yeah. Yeah. It turns out that we've got cell lines. A lot of the cell lines we have do have p53 mutations, and we don't see a difference. We don't see any real correlation between the sensitivity to this drug, whether or not they have a p53 mutation. And one of the things that's exciting to us about this mechanism is that, you know, it potentially would allow us to potentially reduce the doses of other cytotoxic therapy. So in this manuscript that we're preparing with vincristine, Stefan went on to show that we're able to significantly reduce the vincristine dose to get the same level of apoptosis in cell culture. And if we could do that in people, that would be really very gratifying. Yeah.
I have a question.
Yeah.
Any plans for using this drug in combination with radiation?
Well, that's a good question. We haven't done any studies with this drug in combination of radiation, mostly because of just the logistics of radiating cells, and there just hasn't been a, you know, so it could certainly be interesting to think about.
We have unresectable cases where...
Right. No, it's totally a reasonable thing. I'm not sure where I would quite begin to go after that, but we could think about that. Yes. Can't see who's hiding back there, but next to Jeff.
Sure. Thanks. Just wanted to hear your thoughts. Do you think the chemistry community is wrong to think of transcription factors as undruggable, or is there something unique about this target and this drug?
Oh, that's a great question. For those of you who didn't hear, is the pharmaceutical industry wrong about targeting transcription factors? And I would put those in a category, and I spent some time talking about this last year, of intrinsically disordered proteins. And I think they're very wrong. I think that there are potential for targeting virtually any protein in cancer if you think it's an important protein. And there are ways to do it, and the chemistry is fascinating. There is a huge body of literature that shows that small molecules can have impacts on protein-protein interactions. We wrote a review in Journal of Cell Biology in 2014. I was mentored by this fantastic scientist, Peter Wright, down at Scripps, and learned a lot about how small molecules could target protein interactions. And we're really struggling with figuring out how that works in Ewing sarcoma, but the fact that we did it empirically and that we've got this success, I think it's definitely doable. And it's going to be really interesting to see if our drug is successful, and let's hope that it is. Will pharma start to pay attention to this type of thing? Yeah. In fact, just one other, there's a drug that was approved for renal cell carcinoma that affects HIF, which hypoxia-inducible factor, and that's also got disordered elements. Yeah.
No questions?
Great. Thank you.
Thank you, Jeff.